Our next speaker has really been a pioneer. Hans Klebers has been a pioneer in the elucidation of the functioning and regulation of the Wnt pathway, a pathway central to development of organisms and central to the regeneration of cells and stem cell biology in the adult. And Hans is a professor at the University of Utrecht in the Netherlands, and he's the director of the Developmental Biology Institute there. It really was his pioneering work that elucidated the transcription factors that were regulated by the Wnt pathway and the genes that they regulate. And most recently, he's applied all the tools and armaments of the Wnt pathway to understanding the regeneration of our gut cells and in the large intestine, small intestine, which is a Wnt-driven stem cell process that really is one of the prototypical examples of stem cell biology in the adult. And today Hans is going to talk about the Wnt target genes, Lgr5, a marker for adult stem cells in multiple tissues. Hans, it's a pleasure to have you here.
Okay, thanks very much, Arnie, for the introduction and also for the kind invitation. It's my first time to Princeton, and it means a beautiful place, particularly today. It's also, I guess, given the history, a humbling place for any scientist, and that includes me. Yeah, the organ that my lab has taken on to study about 10 years ago is the intestine. Before that, we were immunologists, and actually our immunology led us to clone the T cell factors, these transcription factors that I'll talk about a little bit later. So this organ is really under study. When we made a few knockouts 10 years ago and realized we had a gut phenotype, we also realized that in the literature there were very little tools available, and there were very few labs around the world that were studying this yet. We believe it's, well, these are not my colors. We believe that it's a great system to study stem cell biology and everything that is associated with that. So the small intestine of a mouse or a human being has essentially two compartments, the crypt compartments and the villus compartments. Stem cells have been known to reside near the bottoms of the crypts, and they have mostly been functionally defined. They were never physically identified. It was known that the vast majority of the cells, these light blue cells here, are transit amplifying cells. They divide every 12 hours, and they do so for 2 to 3 days after they generated from the stem cell. And then when they take up when they reach the crypt to villus junction, oh, this doesn't work. Yeah, there it goes. So stem cells reside somewhere here. These are the transit amplifying progenitors. At the crypt to villus junction, it is believed that these cells can take up one of several fates. The dominant phenotype is the absorptive cell, enterocyte or colonocyte in the small intestine and colon respectively. There's about 10% of the cells will produce mucus and are called goblet cells. These are these yellow cells here. Very rare hormone-producing cells, enteroendocrine cells. And then that a fourth cell type, which is unusual in that it turns around. It's presumably born somewhere in the crypt, but turns around and settles at the bottoms of these crypts. And therefore it moves against the flow of all other cells in this system. As I said, these cells will divide for 2 to 3 days. There's another 2 to 3 days before these cells reach the top of the villus, and then they die. So it's an extremely rapidly self-renewing system. Every 5 days you basically renew your entire small intestinal epithelium. This amounts to about 200 to 300 grams of cells every day. So it's massive stem cell and transit amplifying cell activity. This is what by scanning EM one sees in the small intestine and in the colon. So small intestine, again, this is duodenum, has very large villi. These are the associated crypts. You might immediately appreciate that villi are basically fueled by or fed by multiple associated crypts. Up to 10 crypts surround a villus, and it was believed that cells emanating from the crypt, coming out of this little hole here, will move up in parallel ribbons to the tops of the villi, where they will die a little bit later. Colon, molecularly, all our studies show it's almost identical to small intestine, yet pathologists will tell you it's a very different organ. This is the organ where humans get cancer. This is the organ where mice mostly get cancer, but molecularly they're the same entities. Colon has crypts, but it doesn't have villi, it has a flat surface epithelium. And under the microscope, staining for a cell cycle marker, Ki67. On the left, small intestine, in this case mouse. 2 crypts here. Brown nuclei indicate the transit amplifying cells. Stem cells are somewhere here, and these are the differentiated cells on the villus. In the colon, the bottom halves of the crypts have the cycling cells. Stem cells are believed to reside at the bottoms, but have never been seen before. And then, unlike the situation in the small intestine, differentiated cells in the colon are goblet cells and colonocytes at the surface here. They're really mixed in with the proliferative cells and the stem cells. So it's more of a messy organ to study. So our lab tends to try to find genes in the small intestine and then test their functions in both systems, as well as in cancer. The real reason that my lab started working on the gut is the fact that the Wnt pathway turned out to be crucial in gut biology. And we cloned these TCFs a long time ago. It took us a very long time to figure out that they acted together with beta-catenin. And this allowed us and the Bjerkness lab to put the TCF transcription factors at the endpoint of the Wnt signal transduction cascade. And I don't think it needs much introduction, but very briefly, Wnt's are secreted molecules. We have almost 20 of these molecules encoded in our genome. They regulate almost every fate decision during development, but from our studies and from other labs, it turns out that these pathways remain active in particular tissues throughout life. Wnt will bind receptors, frizzled, LRP co-receptor, and this somehow blocks the activity of a complex where for here the most important molecule is APC, a very well-known tumor suppressor in the gut field. This complex in a cell that does not receive Wnt signals degrades every newly synthesized cytosolic beta-catenin molecule, and this is a very rapid process, takes 2 to 3 minutes in our hands. So in a cell where there's no Wnt signals, TCFs are bound up by Gauchos and will repress very specific target gene programs, very tissue-specific target gene programs, I should stress. When Wnt's occupy their receptors, they somehow block the activity of the destruction complex, of the APC complex. As a consequence, beta-catenin very rapidly builds up. So at RNA level, nothing changes, but at protein level, you see beta-catenin within 20 to 30 minutes, reaching very high levels. Some of this beta-catenin probably passively gets into the nucleus, replaces Gaucho, and transiently converts a TCF transcription factor from a repressor into an activator. So this is really a developmental signaling switch. Now, when we found this, we realized that Bert Vogelstein and Paul Pelekes independently had found that in colon cancer cell lines and also in virtually all colorectal cancer cases where APC is almost invariably mutant, as a consequence, apparently, of loss of APC, beta-catenin was very high. And this occurred in the absence of Wnt signals on the outside. And at that time was assumed that this beta-catenin in its second role as a component of adhesion junctions would affect adhesion of cells. Beta-catenin sits on the cytoplasmic tails of E-cadherins on the sides of epithelial cells. But we could actually show that what happens in these cells at the high levels of beta-catenin in the nucleus of epithelial cells find one of the TCF family members, and this is Tcf4, very specifically expressed in adult gut and some other simple epithelia. And therefore, in colon cancer, up to 90% of all cases have high beta-catenin TCF complexes in the nucleus. So we postulated that this somehow reprograms these epithelial cells to behave as transformed cells. And it's probably well established now that the loss of APC and the accumulation of beta-catenin and the formation of these complexes is the principal transforming event that eventually leads to colon cancer in man. And on the microscope, this is what we see. This is a villus in the small intestine of a mouse that already carries a single mutant APC allele. It's called the APC-min mouse. There is a human version of this disease as well, familiar adenomatous polyposis. With high frequency, these mice will develop clones of cells deriving from a single cell where the second APC allele is lost. We stain for beta-catenin here. This is beta-catenin in normal epithelial cells. You might appreciate the accumulation in these lines, in these borders. These are the adhesion junctions. But this is a clone of cells that is a very small adenoma and will continue to grow. They're really undifferentiated cells resembling crypt cells. And eventually this mouse will succumb to the load of adenomas. They can have hundreds of adenomas when they're 4 or 5 months old. We also realized when we were doing these analyses that it looked like there was a little bit more beta-catenin at the bottoms of these crypts. And on a blow-up of this structure here, this is a crypt. These white cells are these are the granules of Paneth cells. We noted nuclear beta-catenin in wild-type Paneth cells. Also, I draw your attention to nuclear beta-catenin in these tiny cells here that we now believe are the stem cells of the system. So it looked like there was indeed some evidence that not only the Wnt pathway was crucial to develop cancer in the gut, but also that at least it was active in the normal crypts. We saw the same thing, nuclear beta-catenin in human colon. In fact, you might see here that this is a nucleus, you go up, there is high nuclear beta-catenin here, and it goes down when you go higher up to the crypt, and these are basically the proliferating cells, and these are the differentiated cells. Again, implying maybe there's Wnt signaling ongoing. We went through all 20 Wnts and we found that at least 4 were expressed by the epithelial cells themselves, a little bit surprisingly, but at the bottoms of crypts, in this case in the mouse. So there are Wnts, there is nuclear beta-catenin. Genetically, when we knock out Tcf4, the mice die at birth. What happens, there is a single phenotypic abnormality. They cannot build crypts, they cannot build epithelium. This is a control mouse, Ki67 stain. So the villi at birth are out of cycle. These prospective crypts with edge birds are still immature. They're called pockets here. They have cycling cells. In a Tcf4 knockout mice, there's not a single proliferative cell in the entire epithelium of the small intestine and half of the colon. So this basically leads these mice to death, indicating that you need Wnt to establish crypts. If we block the Wnt pathway with a soluble protein called Dcop1, a very useful tool, it's a natural gene that binds receptors and blocks them. In an adult mouse, normally we can easily incorporate BRDU in the crypts, so this is indicative of S-phase cells, but in the patches in these transgenic mice where we drive Dcop1, we block Wnt and we lose all proliferation, implying that Wnt is the principal driver of proliferation in the gut. Now, these experiments have often been used to state that to equate Wnt and stem cells. I think in the literature there's very little direct evidence that Wnt actually controls stem cell biology. What it does here, it controls the entire structure, the crypt, and most likely the transit amplifying cells use Wnt signals to proliferate very rapidly. Now, Mark van der Wetering in the lab a few years set out to try to connect those two observations. So on the one hand, there's a very intimate relationship between the Wnt pathway and the gut in terms of cancer. There are probably no or almost no cases of colon cancer that are not initiated by activating Wnt pathway. So it's a very unusual relationship between one pathway and a very common solid tumor. On the other hand, our genetic analyses state that Wnt is an important driver of normal proliferation. It didn't need a lot of imagination to try to connect those two observations. So what Mark did is he took a number of different colon cancer cell lines, and they'll have activating Wnt mutations. So they'll have beta-catenin TCF complexes in the nucleus in the absence of a Wnt signal. But on top of that, they'll have many other mutations, P53, RAS, SMAD4, etc., etc. And then he constructed various inducible ways, always under the control of the tetracycline repressor, so doxycycline driven, either by inducing in stable clones the expression of dominant negative versions of TCFs, or he more recently created vectors where we can inducibly drive siRNAs and we can remove beta-catenin from these cells. And invariably when we do this, independent of the genetic makeup of any colon cancer cell, when we block the Wnt pathway, the cells arrest in G1 within half a day or so, and they don't die, but they stay in culture and they slowly get bigger and flatter, as if they are differentiating. And this was a good starting situation for a microarray that we originally did with Pat Brown, and then more recently repeated in many, many ways, Agilent, Affymetrix, in our lab. And much to our surprise, we found that out of the originally 24,000 features that we screened, there was a very tight program, very reproducible within the same cell line, but also between different colon cancer cell lines, that was downregulated immediately when we blocked the Wnt pathway. Now, these can be direct targets, or they can be targets of transcription factors that themselves are direct targets. We really haven't bothered too much to try to find out how they are regulated by TCFs, but they are all dependent on an active Wnt pathway. And we also saw, obviously, at the other end of the spectrum, we saw a number of genes that were upregulated. Now, when you do gene ontology analyses, all these web-based prediction programs, we basically cannot make sense out of this program. But when we just very simply looked at expression, and that's been the mainstay of all of these studies in our lab, we've gone through all of these genes. We find that the cell, so in this very artificial system where we have a human colon cancer cell line in culture for 30 years, we transfect it multiple times, we add doxycycline, we see genes going down. These genes are physiologically, they are crypt genes. And when we look in human cancers, they really are cancer genes. And one example here is EphB2 was already known to be overexpressed on human colon cancers. This is a tyrosine kinase receptor that's actually involved in exon guidance in the brain. We've shown, I won't go into detail, but it controls positioning of cells in crypts, guided by the Wnt gradient. But this is a small adenoma in human colon expressing EphB2 as previously published by others. But this shows that there is also EphB2 expression in this colon in a gradient in physiological crypts. And this is what we invariably see. So the genes that we find in this very artificial cell line-based system, they are physiological cancer genes and they're physiological crypt genes. Now, when we went through all of these genes in a more detailed way, and we did this on mouse gut, small intestine predominantly, small intestine with adenomas in the APC-mutant mouse, we found somewhat to our surprise that although they were invariably cancer genes, so the adenomas in these guts were expressing the genes, in normal crypts they not always coincided. So the large majority of all Tcf target genes, probably about 250, they will stain these light blue cells that we know are the transit amplifying cells of the crypts. And genes in this program are c-MYC and c-MYP and ATS2 and many, many other cell cycle regulators and proto-oncogenes. So based on that, and not too much to our surprise, we would state that Wnt is a mitogen for these TA, for these transit amplifying cells, as we genetically already knew before we even did this experiment. To our surprise, we found there was a second program that had nothing to do with proliferation, but it marked these yellow cells here, the Paneth cells, and these are terminally differentiated cells that produce lysozyme and defensins and are believed to sterilize the lumen of the crypt, so they will kill bacteria. And these Wnt targets are really mutually exclusively expressed with these ones, and they're late markers of Paneth cells. And based on this, and this is the only thing I want to say about this subject, the same Wnt source not only drives proliferation of undifferentiated cells here, but it also drives terminal differentiation of these Paneth cells at the very bottom. Again, implying that Wnt really does not equate stem cells or stem cell biology. Wnt can do anything depending on the history of the cell that actually is receiving a Wnt signal. In any case, based on this, Nick Barker in the lab hypothesized that possibly hidden somewhere amongst those 250 genes, there were some markers of these elusive stem cells in the system. And I have to stress that when you, in situ hybridizations sometimes give beautiful results, and I showed you a few, but very often you get results that are sort of multiply interpretable. And we had to go very carefully through a number of these genes. We selected 4, and we made a number of knockouts and knock-in mice, and one of them turns out to be a beautiful marker of stem cells, and this is the gene Lgr5, and I'll say a little bit later what it is. The literature, also reviews from my lab, will tell you that stem cells sit at position +4, and this is a long history behind this. There are really very few experiments that back this up, but the reasoning was that in the 60s it was shown that the Paneth cells were not the stem cells to the gut, and that therefore, and these were all proliferating cells, it was therefore proposed that the bottommost cells of this cylinder here were the stem cells, and that then was propagated for 30 or 35 years in all the reviews, and many people will cite many papers, but ultimately there's only one proposal in the literature. It has been claimed that these cells retain DNA labels, yet it also, the same groups will tell you that these cells cycle heavily, and we've never been able to show actually that these cells retain labels. Paneth cells will do so because they're terminally differentiated, and they have a long, unlike all other cells in the system, they have a very long half-life. They can live up for 2 or 3 months. Lgr5 stained, this is a mouse I'll show you later, so we stained for protein. It stained very strangely shaped structures that sat in between the Paneth cells. And then when we went back to our Ki-67 analysis that we've done many, many times and really missed this observation here, we noticed that in between Paneth cells there are cells with very elongated nuclei that basically don't appear to have any cytoplasm. So they're just a big nucleus squeezed in between two Paneth cells. Very differently shaped nucleus, but invariably they are Ki-67 positive, which really doesn't point to them being stem cells, because you would assume that these cells would be quiescent. Lgr5 is an orphan G-protein coupled receptor. There's some literature on it. It presumably signals like its relatives, the LH receptor or the FSH and the TSH receptor. Like these receptors, it has a large extracellular leucine-rich repeat domain, predicted to bind to a cysteine-not ligand. It's not. We actually have a few candidates at the moment, but no definitive evidence that they will bind. Lgr5, interestingly, sits in a small subfamily or clade of 3 highly related genes, 4, 5, and 6. There is a fly homologue of Lgr5 over this subfamily, and for the fly homologue, there is a ligand known, it's called bursican, and it consists of a heterodimer of 2 cysteine-not proteins. So we're actually looking to find the equivalent of that in human and mouse. The first mouse that Nick obtained was a LAG-Z knock-in, and this was to study expression. This was when we still had 4 candidate stem cell markers. This mouse inserts an IRS sequence and then a LAG-Z reading frame upstream of the 7 transmembrane regions, so it will cause a null allele. While we were doing this, Morita et al. Published that an Lgr5 mouse is neonatal lethal due to some abnormalities in the lower jaw and the tongue, by which they cannot swallow, they cannot drink milk, and then they die. However, we could use our mice in the heterozygous situation to carefully document expression of the gene. Well, it's all over the place, so developmental biologists get very excited. And although I'm heading at the Developmental Biology Institute, I have no clue what these structures really are. Most importantly, that's where we got very enthusiastic, is that all this expression disappears around birth, and then we're left with very few cells in the adult mouse that keep on expressing the gene. And this is the small intestine. These are the Paneth cells again. The nuclei are here in red, and this is one of those cells, is another one. And we estimate there are probably about 6 of these cells at the bottoms of the crypt. So they're tiny cells. They're probably 20% of the, or 10% of the volume of Paneth cells, but they are real cells. At least they have nuclei, and they incorporate the BRDU, etc., etc. So we then studied carefully if there were any quiescent cells, and if we give continuous BRDU label to a mouse for 24 hours, in essence, we label all of the cells that we can, all of the CBC cells, as they're called, crypt-based columnar cells, at the crypt base. So it doesn't look like there's any of these cells is not in cycle, which is, again, wasn't pointing to a stem cell-like phenotype of these cells. So Nick then created another knock-in mouse, and in this case he inserted in the first exon 2 proteins, the well-known enhanced GFP, so this is a stable version of the GFP protein, and that gets to be important a little bit later that it's stable, an IRS sequence, and then behind it he inserted an inducible version, probably well known here, of the Cry recombinase. A single shot of tamoxifen will activate this protein. It's in this version, it's bound up using the estrogen receptor fragment that's associated here. It binds to the heat shock protein 70 in the cytoplasm. Tamoxifen will release it from that cytoplasmic anchor, and then transiently Cry will be able to recombine LOX-P sites in the nucleus of the cell that expresses the protein. So the idea would be, and I'll get back to this, that we'll create a mouse where every Lgr5 cell will be green and will have a latent but activatable version of the Cry enzyme. And I'll tell you why we made that mouse in that way later. So it works beautifully. This is maybe a bit difficult to see, but these are 2 large villi in the duodenum of a mouse. So this is the region where the villi are biggest. These are a number of associated crypts blown up here. And again, we see probably 6 cells of these tiny cells that have the GFP protein. This is live tissue, so we can actually follow these cells while they are dividing on the confocal microscope. It looks like they don't divide asymmetrically. We also see there's a little bit of carryover of GFP protein to what appears to be the immediate daughters. And again, I'll get back to that, but the rest of the crypt is really negative. And we now know from many other experiments that this is a black and white, or I should say a green and black marker for live stem cells in the gut. Peter Peters in Amsterdam helped us with some EM using the GFP that we had now expressed in these cells. We could use Immunogold labeling, maybe a bit difficult to see, but this would be the signal that we get. This is a Paneth cell here. The crypt points up and to the right, so this is the crypt bottom. This is a Paneth cell. These are Paneth cells with those granules that have defensins and lysozyme, etc., etc. This is one of the CBC cells, so it touches the lumen here. It has even some microvilli. There's a tiny thin stretch of cytoplasm going all the way down, and then it has a broad base in one direction. The nucleus outlined here fills up almost the entire cell, so it really looks like a genome replication machine and nothing more than that. There's a few ribosomes, but a few mitochondria, but very few. And then at a 90-degree angle, it really is a very, very flat cell. So this really looks like a pizza slice and not much more than that. You can see the size of this cell relative to these huge Paneth cells around it. So having seen that our GFP is expressed in the cells that we hope them to be expressed, Nick then utilized the other protein that we knocked in, the inducible CRE protein, in an attempt to genetically mark the cells and then follow their offspring. Because when the stem cell divides, a GFP positive cell divides, it'll give daughter cells. If they no longer express the Lgr5 gene, they will no longer be green. However, if we have marked the cell genetically, we can probably follow its offspring. This is a often used trick in developmental biology. It's a mouse. This particular mouse is made by Phil Soriano. It has in the Rosa 26 locus a strong promoter, a Lgr5 reading frame, but in between those two DNA fragments is a stopper sequence or roadblock sequence that is flanked by two LogSP sites. So this mouse is entirely white, but if a single cell expresses a CRE enzyme functionally in the nucleus, it'll excise the roadblock, and from then on that cell will be blue. But if the cell divides, also its offspring will be blue forever. So it's a genetic mark, unlike the GFP marker that's only in the cells themselves, but is lost in the offspring. And the idea would be is we cross these two mice, we let them grow adult, they will be entirely white, but if we now shoot a single low-dose tamoxifen into these mice, we will mark the stem cells and can study what they do. So before shooting tamoxifen into the mice, we'll have green stem cells, but there's no blue in the system. But when we give a little bit of tamoxifen, and I stress, we give very low doses, so we don't induce a lesion in all of these cells, but we only induce a lesion in 1 out of every 10 crypts in one stem cell. So we then create clonal offspring, and we can see what the clone actually contains. And this is then what we hope to see, so a blue stem cell, and then over time, and we would predict that this would take about 5 days, we would create a blue ribbon of cells, and then from then on, this would be sort of a clonal conveyor belt, because the stem cell, if it's a real stem cell, would stay alive, and every day would divide, as we knew, and every day would put daughter cells in place that rapidly expand, and then create the continuous conveyor belt for, hopefully, long periods of time. Now this works beautifully if we delete, and so if you inject tamoxifen and we wait 12 hours, we see, this again low dose, so we see single stem cells in 1 in every 10 crypts or so in the CBC position. 1 day post-induction, we typically already start seeing larger cells a little bit higher up in the crypt, so we believe these cells have already gone through 1 division, and we're actually seeing a stem cell and a daughter cell. 5 days post-induction, we get these ribbons. 60 days post-induction, we still have ribbons, so now the epithelium has renewed 12 times, and the ribbon is still there, so the same stem cell is still dividing every day and producing offspring. I should also stress that every time we see a blue crypt, 5 days later, it has created a ribbon. So we don't see any evidence that we are marking quiescent stem cells. It looks like every stem cell we mark is a rapidly cycling stem cell. We've now waited longer, so a year after induction, and this is whole mount. Here you look on the outside of a gut. The muscle is very thin, so we can see the blue stain through the muscle, and these are the bottoms of crypts. Often we see little groups of crypts now together, and it's known that crypts can, when there's damage or when the mouse grows, It can actually undergo crypt fission, so it divides sideways and creates two crypts, and I think that's what's happening in these positions here. Here's another version. And this is a side view, so this is the muscle layer. We cut through the epithelial layers. These are the crypts, and these are the villi. And you can see there is a high frequency. Actually, the frequency of blue ribbons remains unchanged when we follow the mice from 1 day to a year, implying that, yes, these are cycling cells, but they manage to be there for at least a year, and probably for the lifetime of a mouse. And this means that they undergo in a mouse probably 700 cell divisions, and if we have the same system in our guts, we have to have cycling stem cells for 80 years. And the challenges in terms of telomere maintenance and genome integrity maintenance are I guess, enormous. Now, are they, so they're long-lived, are they pluripotent? They are, because in these clonal ribbons we see all the cell types. These are goblet cells, these are absorptive enterocytes, these are Paneth cells, and these are endocrine cells. We can sort them out, so we can isolate crypts. It's very easy. You just strip off the villi when you open the gut, incubate briefly with EDTA, and then these crypts float out. Here you see the stem cells. We can dissociate them with a single step of trypsin on the fax. We can sort out pure stem cells. On a blow-up, you can see it appears there are two populations, and we have good evidence, I'll show you a bit of that these are the real CBC cells, and these are their immediate daughters, and these are all the later daughters that no longer have GFP. Here you see the cells under the microscope. A single mouse will give us about 500,000 to 1 million of these cells, so and we can do it in 2 or 3 hours. So it's a very rapid procedure that gives us, in a single step, gives us essentially completely pure stem cells, which I don't think exists in any other system. When we do a race with comparing these cells with the bottom cells, it's a little bit of a messy array because there's lymphocytes, macrophages, Paneth cells, many other cells here, which makes it difficult for the computer to interpret the data. But when we compare these stem cells with what we believe are the daughter cells, we get about 100 gene differences between small intestinal stem cells and daughters. We've done the same thing in colon. There's an enormous overlap. It's 80%, so there's very few small or colon-specific stem cell markers. The top gene in all of this we make is Lgr5 itself, implying that indeed the cells we are sorting out are the Lgr5 positive cells, and the ones with slightly lower GFP are the daughters, and they somehow have managed to downregulate Lgr5 message. Incredibly, it's basically absent in the daughters, and it's fully present in the there were some other, I won't tire you too much, but some other genes on that list that we already were working with. This is a Wnt target, Akits-Cute homolog 2, or MASH2 is the other name. This was actually a new marker for these stem cells. Just a few slides. MASH2, Laurens van der Vlier in the lab had already been working on this, made a monoclonal, so it does stain these crypt-based columnar cells, the nuclei of those cells. He made an inducible knockout. Igor already mentioned that MASH2 is a trophoblast marker, and knockouts, nulls, will die halfway gestation because they have no placenta. So we made it, and it's an imprinted gene, which complicates it further. But we have a floxed allele, and when we delete it, the other marker, so the gene itself is gone, but the other marker I just mentioned, and I won't go in any detail here, olfactomedin, is in fact a beautiful marker, incredibly strong for our stem cells. This is a blow up here, but these are the walls of the gut, and you see how strong the signal is on in situ hybridizations, and these are the individual CBC cells. When we knock out MASH2, we basically lose the signal entirely. And we've made transgenics, and it looks like MASH2 is the or one of the master regulators of the stem cell phenotype. So if we express MASH2 on villi, all of these villi cells look like crypts and stem cell-like cells. And actually the mice will develop a lot of transformed cells and eventually cancer. So this works, and I think we are in a situation of isolating pure stem cells, study their gene expression profiles, their microRNAs, which we're doing much like we heard from Kurt. We are actually together with Steve McKnight looking at their metabolome. They have hardly any mitochondria, and from all the enzymes that we see, they avoid producing oxygen radicals. So probably they're trying to not damage their DNA. They have very high levels of DNA repair enzymes. They have very high levels of BCL2. So the cell is really poised to proliferate constantly, but also to protect its genome as much as possible, which theoretically one would already assume. Well, so to summarize this bit, I think we definitively defined a cell in the small intestine, the crypt-based columnar cell, that had actually been proposed to be a stem cell 40 years ago by LeBlanc in Canada, and that this cell makes, over very long periods of time, makes all the cell types of this particular epithelium. And importantly, it is a very rapidly cycling cell, very, very counterintuitive. Now, in the colon, we find the same thing. So the same mice have green cells in the colon crypts here, as you can see here. And again, using the tracing trick, the Rosa 26 trick, we can label a single cell. And then when we wait 5 days or 60 days, we have labeled entire crypts. Again, all of the cell types are blue. And again, when we wait a year, they are still there. So these cells not only are the small intestinal stem cells, they also are the colon stem cells. Stomach, where there's a lot of controversy where the stem cells are. It's not a Wnt-driven epithelium. It's very different. It makes hydrochloric acid, as you know. It makes pepsin and various other intrinsic factor. There is, as you can see here in this whole mount, there is a Lg5 at the bottoms of the epithelium, and they blow up. You see it here. These are the glands. Stem cells in the textbooks are believed to be in the middle of the glands at the isthmus, yet we see cells at the bottoms of the glands that express the gene. And in the same mice that we used for our tracing, this is duodenum. So here we labeled stem cells, and as I showed you earlier, we labeled for long periods of time stem cells on the villi. This is stomach epithelium. At day 1, we will label cells at the bottoms of these glands, but a few days later, the entire epithelium is labeled, and this again stays forever. So also in stomach, although we don't think it's driven by Wnt, it's probably another way by which the gene is controlled. Lgr5 appears to mark unique stem cells. In the hair follicle, I don't want to say too much, but Elaine Fuchs and George Kotsarelis and Fiona Watt have shown that the bulge region in the hair follicle resides somewhere near the sebaceous gland, harbors quiescent stem cells. We find in collaboration with Rune Toftgaard in Karolinska in Sweden that Lgr5 seems to mark cells at or near the bulge, yet they are all proliferating. They don't seem to be descendants of the quiescent cells. They're really an independent cell pool. This is another GFP knock-in experiment. So this is skin. So you see telogen hair, so it's hair that's not growing, and these are anagen hairs, and you see a trickle of stem cells. This is where the hair is produced. So this is where all the proliferation takes place. The transit amplifying compartment, that is negative, but stem cells, or at least our Lgr5 cells, sit all along the follicle. And again, if we look in our mice and we wait for long periods of time, up to a year, we can produce blue hairs that will stay forever and go to many, many hair cycles. So again, this is a rapidly proliferating cell population. It's not quiescent. If we label it in the tracing, they will make hair forever. Very different from the quiescent cells. So in the bulge, we can show that the two populations live side by side. There is a quiescent population, and there is our population. It looks like in normal circumstances, these cells are the ones that really make the hair follicle. So based on this, we would state that Lgr5 marks multiple types of cycling adult cells, but not all. There's a number of organs where Lgr5 is not expressed, like the prostate or the lung, muscle. We see it in retina and brain, and in a large number of other tissues, memory gland, as I'll show you. However, I already told you there are 3 genes in this family. We've continued to make all the knock-ins for the other 2 genes. So we have a Lgr5 knock-in, and we have the GFP-KI knock-ins. Some preliminary data. This is the memory gland, where we already know that basal cells are positive for Lgr5, and they have the exact phenotype that stem cells should have. We have done no tracings yet. Lgr4 is in the same cells as is Lgr6, and we see this in many tissues that they are co-expressed. Sometimes 2 of the 3 are expressed, sometimes only 1. But based on this, another postdoc in the lab, Andrea Hagebart, has made similar sort of GFP-KI-RT2 knock-ins in Lgr4 and Lgr6. We just started to analyze them, but some preliminary data from the Lgr6 mouse, again in the hair follicle. I showed you, this is the hair, this is the sebaceous gland. The bulge is somewhere here, and with Lgr6 we find cells that are higher up. we sometimes see these cells getting out of the hair follicle, so they go in the opposite direction. And it looks as if this is, again, a cycling population of cells that doesn't build the hair, but it builds the epidermis. And we're now doing the Rosa 26 experiments to prove that. You see Lgr5 here is much lower. There is a sebaceous gland here. Lgr5 sits here, so it sits below the sebaceous gland. And I expect when we look at our in situ and our LAC-Z analyses that probably all adult tissues will have small numbers of cells that express at least one of these genes. So implying that they probably the gene family will mark in every tissue its cycling stem cell population. And these are then the conclusions to here. I have a few words, if I have time, Arnie, over the roles of these Lgr5 cells in cancer. In the gut, I already told you that APC is lost, and this leads to the formation of adenomas, and many more mutations then create adenocarcinomas that can be invasive and metastasize. Lgr5, as I already showed you, is expressed in these small cells at the crypts, unlike most other Wnt targets, but also in adenomas, and that's why we originally missed it. It's not expressed everywhere, but it's expressed in very rare cells on this in situ. If we try to document how Lgr5 expression develops over time in a tumor, this is a small adenoma in an APC-mutant mouse that we can recognize with the beta-catenin stain. It sits at the bottom of a villus here. When we stain for Lgr5, we find that all of the cells are positive, as are the stem cells that are sitting here. But if we now let that tumor grow, and we come in a few months later, we have a large tumor here. Well, these dark fields, again, are the transformed cells, so there's a lot of normal tissue surrounding transformed tissue. Now expression is really gone back, and we've found at the most 1% of the cells in that tumor expressing Lgr5. Now we're trying to, with our tracing techniques, we're now trying to label these cells genetically in the Rosa 26 system and see over time that they in situ actually behave as stem cells. This is a blow up here. You see that this is all cancer tissue, but there are these rare cells. Often they have the appearance of the stem cell, this very slender shape, as if they're sort of the tumor itself is trying to rebuild its stem cell compartment. Then we irradiate these tumors, we kill most of the cells, but actually these cells stay behind. And that's one part of the cancer stem cell hypothesis, that the cancer stem cells that we might be looking at here are actually more resistant to chemotherapy and radiation than are the bulk of the proliferating cells in the tumors. So this is still a bit preliminary, but I think given that Lgr5 is such a good marker for normal tissues, it's probably going to be a very good marker for cancer stem cells in the tissues where Lgr5 also marks normal stem cells. This I probably don't have to show. Another issue, I think, where stem cells come in cancer is what is actually the cell that sustains the first oncogenic hit. And we can actually ask that question in our system. We have a floxed APC allele that we can delete with a P450 CRE, is an inducible intestine-specific CRE transgenic, which at will, upon injection of beta-nephthoflavone in this cross, we will delete APC, and this is done mostly by Owen Sansom in Dundee, with some help from us. And what that does is it actually transforms acutely. So this is an adult mouse before removal of APC in the epithelium and after removal of APC. This is just 2 or 3 days. And what happens is that where normally you only have cycling cells in the crypts, with lead APC, basically all of the cells of the crypts will keep on proliferating, move up the sides of the villi. By day 3 or 4, they have reached the top of the villus and the mouse dies. So you basically transform all cells in the system. But we really don't know from this experiment what the cell is, the identity of the cell that sustains the first hit. And to do so, we can now use our stem cell specific CRE and do the same experiment and compare it to a low dose P450 CRE that we give orally. So now the beta-nephthoflavone comes in, it deletes up to about half of the crypt, it deletes on the villus, but under these low dose oral treatments, we almost never reach the bottom of the crypts, and we know this from tracing experiments. Okay, so this is the experiment. So we have a mouse, floxed APC, we give beta-nephthoflavone. What we do is we get a lot of transformations. You can see here, these are beta-catenin, positive small adenomas or microadenomas. What we actually see is that these will never progress. So they sit, for instance, here at the top of a crypt. There is nothing lower in the crypt, as we had hoped to see. This adenoma, if you come in 2 or 3 months later, is still there. It's P21, P27 positive. It somehow is transformed, but it apparently goes through a few rounds of division, and then it stops. Implying that if we take APC out of the middle of the crypt, out of transit amplifying cells, we at least in this system, don't make cancers. The other ones on the villus, here, this one probably lost the APC while it's getting out of the crypt. It divided a few times, got stuck on the villus. It is, or is it slightly changed, but it is Ki67 positive, so they tend to proliferate a bit, but then they're lost at the top of the villus and they're shed into the lumen of the gut, and they don't make tumors either. If we now do the same thing using the stem cell specific so now we specifically delete APC in stem cells in this system. At 3 days, we start seeing beta-catenin positive stem cells in the crypt. So at 1 day, we get we give low dose tamoxifen, a single stem cell that has a high in beta-catenin, so is APC-mutant. At 3 days, we see that these stem cells have sort of sideways, they have multiplied. They're now 3 to 6 in the crypt. But then this process stops, and then we see these things forming. So adenomas higher up in the crypt, but the stem cells remain here. And by any other stain, we would not recognize that these are transformed, that these are APC-mutant stem cells. So they look like physiologically totally normal stem cells, but meanwhile they'll build up an adenoma higher up. And then when we wait 8 days post-transformation, we still have the stem cells at the bottom of the crypt, so now we form the much larger adenomas. I just saw the most recent data where we wait 3 months and we get huge adenomas, but still the associated crypts will have stem cells that look like they're normal stem cells, but they really are, they are transformed because they have lost APC. So based on this, at least in this system, we would postulate that the easiest way to become a cancer cell is to start with a stem cell. And apparently also in the earliest forms of a tumor, so in these microadenomas, there already is a hierarchy. So there are stem cells that actually stay at the stem cell position, and their offspring produces a tumor. So there are already 2 cell types, stem cells and transit amplifying daughters, yet both of them are transformed. And I think with that, yeah, the conclusion is that, as I already told you, these stem cells are the most likely targets for the oncogenic event in the gut. It's possible that this holds in many other systems, but one needs really a Cree that is stem cell specific to do these experiments. I'd like to end and mention a few of our collaborators. The hair follicle data that is just resubmitted was mostly done in Rune's lab using our mice. Peter Peters and Pekka Kujala helped us with the EM and are still helping us with the EM of all these stem cells. The APC flux experiments we do in close collaboration with Owen Sansom. Ongoing memory gland studies we do with Jos Jonkers in Amsterdam. And the Lgr5 work was almost single-handedly done by a British postdoc in the lab, Nick Barker. And the more recent work on Lgr4 and 6 was done by another German postdoc, Andrea Hagerbart, in the lab. And with that, I'd like to thank you very much for your attention.
So there are questions? Maybe I can ask one, and then we can get the microphones around. The stem cells are constantly dividing under normal conditions, and so you'd think that they might have telomerase. Because if they rejuvenate every 5 days, the whole phyllis, they're going to run out of time and space unless they can rejuvenate the ends of their chromosomes. Have you looked for telomerase activity in that compartment?
Well, we haven't done it, so we can now, I guess, isolate enough cells to look for telomerase activity. We've studied telomerase length, and it doesn't change very much when you go from very young mice to 12-month-old mice. But yeah, the expectation is that telomerase is there in large quantities. Yeah.
Thank you for an excellent talk. Just a general question: is there Lgr5 genes or like genes in Drosophila and in zebrafish? And are you investigating them?
So zebrafish, the Sanger database gives 2 genes, an Lgr4 gene and 1 gene that might be the Lgr5, Lgr6 precursor. Yet we know that the sequence is not really curated well, and we're still looking for the real Lgr5 homolog. We are making mutants of. Our institute set up the stilling method, so we can make by reverse genetics, make zebrafish mutants, and it looks like we have all 3 of them, or at least all 2 that we found mutant. Fly it was on the slide, have a single gene is called Drosophila Lgr5, and so the mutant phenotype is I'm not really sure whether it has anything to do with stem cells, but they fail to. It's a pupal phenotype. They fail to pigment their exoskeleton and to inflate their wings. The ligand is known for Drosophila Lgr5, and it was found actually. It was found so the receptor is in thoracical segments, but the ligand came from the head. And the way it's done is that people take larger flies, blowflies, and then tie a hair around their necks, so they look. The figures in that paper look very strange. And then they basically turn into a reaction vessel to find the ligand. So it's a Japanese lab that made extracts from heads of flies, injected them into the bodies of these flies where the necks were tied up, and purified the ligand in that way. So when they injected it, the wings inflate, and they have this rope around their neck. And it's a cysteine dimer encoded by 2 different genes, which makes expression cloning very difficult. So we're now biomology trying to find. We have a few weak binders, but I'm not convinced that they are the ligands.
Hans, as you beautifully described, the Lgr5 and the other Lgrs identify a cell which has the properties of a multipotent progenitor, doesn't have the quiescence and some other properties of something you'd naturally call a stem cell. So I wonder, if you go to a system like ESLs or like hematopoietic cells, where you would find the LGF, in the multipotent stem cell, or in the multipotent progenitor, or in the stem cell, or both?
Yeah, so we don't see Lgr5 in the bone marrow, nor do we see it in the ESLs. Expression of the genes is low, so it's difficult to detect anyway. Lgr4 and 6, I'm not really sure. I've looked at the ES databases. By PCR, we see them in the ESL lines. By looking at array databases, I don't find them. So we're waiting for the Lgr5 knock-ins. We're doing early embryos to see whether they really stain and what the cells are.
Well, just echo, it was just a beautiful talk, great use of contemporary technologies. I was interested in the ligand a bit because of 2 reasons. One, this intimate relation between the Paneth cells and the cycling cells, and whether that's required or at all important. If you can ablate Paneth cells, does anything happen? May they be a source of ligand? And 2, also being able to sort these cells, can you look at the cysteine knot, whether it's local, paracrine, autocrine, or as you said, from the blowflies, quite distal. And then the last question is just with respect to the skin, the Frieda-Miller skip cells, do they express Lgr5 or are they devoid of it?
Well, to start with the last question, I don't know what these cells are.
Me neither, but I thought maybe that would be one way to
so what we
characterize
see
them.
with the Lgr6 Lgr5 mice is we see this small cluster of cells in hair follicles, and in some areas of the mouse, again, Lgr5 is a stable protein, we see that it trickles out of the hair follicle and spreads in basal cells around the hair follicle. And based on that, it's like a tracing, but it's not a genetic tracing. So based on that, I assume that what these are, they are sort of the ultimate epidermal stem cells. As it's known that hair follicles can rescue skin when you ablate the rest of the epidermis. But I don't know whether they are the cells that you named. Because what cells did you ask?
Frieda-Miller has published the so-called skin stem cells, which he calls skip cells, and shows that they go on to give rise to a large number of different cell types.
Yeah.
Even neurons,
Yeah.
in your publications. So in trying to distinguish between the Frieda-Miller cells and Lane-Fuchs cells, I was wondering how that's parsed with respect to...
So I think the Lgr5 cells are much like Elaine Fuchs's or George Kotsourelis' cells, who described them first. The Lgr6, we just started working on it. Then your first question, the Paneth cells, there is a genetic model that we were involved in studying, the SOX9 knockout. SOX9 is again a Wnt target, has no Paneth cells. What happens is that the entire niche fills up with stem cells. So you end up with 20 or 30 rather than 6, implying that the number of stem cells is really defined by the niche. And your second question, so the ligand, yeah, so we have one colon cancer cell line that expresses Lgr5. We believe that it might have the machinery to signal, and we're using that cell line to, and that's actually, we're very lucky, the cell line we initially took to find the Wnt target gene program is this LS174. And it now looks like that particular cell is identical to our stem cells, whereas all other colon cancer cells are more transit amplifying like. And LS174 is a beta-catenin mutant. It's not an APC mutant. It's diploid, so that's why we really liked it for our arrays.
Hans, that was a great. Based on the microadenomas,
I can repeat.
check. Thanks. Yeah, it's hard to. So based on the microadenomas that you showed late in the talk, is it feasible to propose a transformation mechanism by cell-to-cell contact that the cancer stem cells are not actually part of the tumor, that they reside far from the adenomas and they influence a progenitor or other type of cell for transformation and tumor development?
Well, so I think this is kind of a tracing experiment because we delete APC in the stem cells. We can visualize that by staining for beta-catenin. And then the adenoma that forms is high in beta-catenin, so it also has lost APC. We're now including the Lgr5 reporter to show that, but I think they are really the same clone. So it starts on day one with one stem cell, and although the adenoma ends up higher up than the stem cells in the system, I think they're the same clone, much like what we normally see in the crypts. But I think the definitive experiment there would be the Rosa 26 co-cross.
So I have two questions related to the discussion you just had here. And first, in the APC-mutant mice, you showed the Lgr5 could be possible cancer stem cell marker. I just wonder whether you look at the beta-catenin staining in those tumors, whether that, if it's really Wnt expressed, decide the expression of Lgr5 or other regulation might come in if it's not totally correlating with beta-catenin staining.
So we've looked at beta-catenin because levels of beta-catenin, nuclear beta-catenin, can differ within subsets of cells and tumors. But we really don't see why these cells, the cells that do, why they do express Lgr5 and why others don't. It looks like, I tried to show that, but they reside in regions that resemble normal crypts in these adenomas. So the tumor might recreate niches, and because we don't know why in a normal crypt Lgr5 is only in those cells where it's a Wnt target. So we think there's something to the niche that helps the expression of Lgr5 relative to all other Wnt targets. We don't know. It's definitely not the high beta-catenin.
So the second is in your APC and deleted stem cells. I wonder whether you compared with the properties with normal stem cells, whether they're proliferating faster, or it's just because normal stem cell already have activation of Wnt. So if you knock out APC, will not have much effect on it, due to also some tight regulations of other signaling pathways.
I think this is a good question. So we haven't really studied this. We just have done 5 or 6 mice. But what we do see is that the numbers of stem cells immediately increase, of transformed stem cells immediately increases enormously at the cost of the normal stem cells. So it looked like they occupy the niche where they're because we only hit one stem cell. A few days later we have 4 or 5 or 6. It also looks like they spill over in the next crypts, as if they really are, or either they're doubling crypts or they're moving into other crypts. So there is definitely, they somehow win from the normal stem cells. Yeah.